everybody. Today we're going to take a look at the flow of genetic information. We've been through many different parts of the cell. We've learned what those different parts do, but ultimately what we had was a parts list. So now it's when we go from the parts list from within the cell to actual working plans. How do those parts come together to do something meaningful? And in this case here, what we're going to be looking at is how DNA goes from this simple molecule you see here into working proteins of an organism. We'll start with talking about the flow of genetic information by way of what's called the central dogma of biology. And how it works is like this. We know that each of the cells contained an entire library of DNA for that particular organism. So it contains the entire parts list and the working plans for how that organism, and let's get down even smaller in size, that actual cell will perform. So we start with the DNA, and that DNA is going to be changed into RNA. The RNA will eventually become a working protein. So essentially our central dogma of biology then is from DNA, we go to RNA, and then to protein. And for nearly the entire realm of biology, it's going to work this way. When we take a look at viruses a little bit later, we'll see some rare occurrences where it doesn't necessarily go in this direction, but the exceptions to the rule are minute compared to the general nature of this flow of information. Now, what we'll look at today is we're going to focus on two particular processes. We'll have a process, we'll just call it for now process 1 that happens here, which takes you from DNA to RNA, and then here we'll call it process 2. Both of those processes are going to be key in going from DNA to protein. The informational content of DNA is in the form of specific sequences of nucleotides called genes. So we've learned that along stretches of DNA, some of it may code for something very important, and we call that a gene, something that can be passed on to a future generation. Now, if we get a little bit more basic than that and we talk about what exactly is a nucleotide, well, hopefully it's review at this point, but a nucleotide is a nitrogen-containing base, a sugar, and a phosphate. So you should definitely look at the structure of DNA before you go into learning how these processes work. Having a working knowledge of the parts of DNA will help you immensely as you try to go through these two processes. Now, the gene is something we said that can be inherited from generation to generation, but the definition of the gene actually keeps changing a little bit each year as we learn more and more from the findings of the Human Genome Project and proteomics. We then start understanding that the definition for genes is really hard to pin down, and it will continue to change. But basically, we're talking about DNA that is inherited by offspring. It's going to lead to specific traits by dictating the synthesis of proteins. So overall, we'll have a collective term that describes that, and probably the best one would just be gene expression, the process by which DNA directs the synthesis of proteins, or RNA. We learn more about RNA year in and year out, and we find what a crucial, pivotal role it has in cellular biology. Since RNA seems to be the stop after DNA, and we get there by way of a process, let's talk about RNA first. RNA is an intermediate between genes and the proteins for which they code. And RNA is different from DNA. So with RNA, we saw that uracil will replace the nitrogenous base thymine. RNA is also single-stranded compared to DNA, which is double-stranded. RNA has a different form of sugar than DNA. So if you recall, when you studied the differences of those two, of nucleic acids, we saw that RNA had a sugar called ribose, and DNA has deoxyribose. So you can see a key part of their name comes from the sugar, which makes up part of their general anatomy and structure. Here's a nice view of the two of those molecules, and the thing that sticks out, or should stick out the most, is the fact that this here is your double strand, which makes it DNA, and this one is single strand. So just by eyeballing the two strands, you know which one is which right off the bat. Here we have our 4 nitrogenous bases. We have cytosine, guanine, adenine, and thymine. So remember, our single ring structures here are pyrimidines. I'll put PYR for those. And then our double ring structures are our purines. But in the case of RNA, you can see that our T, our thymine, is replaced by uracil. Still a single ring pyrimidine, but it will replace everywhere where we find a T on the DNA molecule. There are 3 classes of RNA. The first class is called messenger RNA, or just simply mRNA, and it takes a message from DNA in the nucleus to the ribosomes in the cytoplasm. The second class is tRNA, or transfer RNA, and that's going to transfer amino acids to ribosomes. And third, we have ribosomal RNA, or rRNA, along with proteins will make up the ribosomes, where the polypeptides will actually be synthesized a little bit later. We'll see each one of these in action in the processes to come. There's 2 major steps in synthesizing a protein. So you can see here we have a couple of different types of cells, so that let's kind of differentiate what's going on here. This is a bacterial cell right here. And this is going to be our eukaryotic cell, and from its shape, we can probably be safe to assume it is a plant cell. So we're going to see a lot of similarities in these 2 processes between prokaryotic and eukaryotic, but there's also some key differences too. So the first process we'll mention here is called transcription. Transcription is where DNA serves as a template for RNA formation. Okay, so in our prokaryotic cell, we can see that transcription happens inside. We know there's no defined nucleus in a bacterial cell. But inside of our eukaryotic cell, we can see transcription does happen within the confined nucleus. And what's going on here, and you can see it from the diagram, is that your double strand here is, you can see producing a single-stranded mRNA from it. And you have the same exact thing happening here in a eukaryotic cell. I'm kind of a stickler for locations of where processes happen. So in the case of the bacterial cell, we have the location of the cytoplasm as our transcription location. And so let me draw a big arrow right there. And then in a eukaryotic cell, the transcription location is within the nucleus there. Now, that's only half. That's the first process, or process 1 I alluded to in an earlier slide. The next process is called translation. That's process 2. And that's where your mRNA transcript, that copy from your template, directs the sequence of amino acids in a polypeptide. So you can see in the prokaryotic cell, that is going on right here. You see a polypeptide being produced. You see a ribosome in action. You see the mRNA seemingly being thread through that structure, and that's what we call translation. So that also occurs inside of the cytoplasm of the bacteria. In a eukaryotic cell, however, we see that translation leaves the nucleus and it happens out here in the cytoplasm. So during translation, we see that occurs in the cytoplasm of a eukaryotic cell, but in the cytoplasm of a eukaryotic cell and the cytoplasm of a prokaryotic cell. Now, there's one box that's kind of left untouched here, and that's because there's some added extras that have to be done as before translation can happen. So your transcription, your mRNA, must be changed, tweaked just a little bit in order to be properly transferred out of the nucleus and copied properly. And that's called RNA processing. And you can see where here you just have your red mRNA. Here you have the same thing, but you got a couple little extras here. You got this yellow tip on this end, and you got a yellow tip on that end, and more to come on that. Okay, next on the agenda, we need to talk about the genetic code. And we have a question to ask. How are the instructions for assembling amino acids into proteins encoded into DNA? There are 20 amino acids, but there are only 4 nucleotide bases in DNA. So, how many bases then correspond to amino acid? If a single nucleotide coded for an amino acid, the most you could have would be 4 amino acids. And we know that's not the case. We found 20. If 2 nucleotides coded for amino acids, the maximum number of combinations would give 4 to the second power, or 16 amino acids. Still not quite enough. But if 3 nucleotides code for amino acids, then you can have 4 to the third, or 64 combinations of amino acids, more than enough for the 20 amino acids. Now, is it too much? The answer is no, and we'll see why in a minute. So, the existence of something called an codon is going to allow us to have 64 different combinations of 3-letter combos of these nitrogenous bases, and it will allow us to cover all 20 of the amino acids. Let's learn how. The flow of information from a gene to protein is based on what we call a triplet code, a series of non-overlapping 3 nucleotide words. These triplets, called codons, are the smallest unit of length that can code for all of the amino acids. If we looked at an example, any 3-letter combination of those nitrogen-containing bases will code for a particular amino acid. So in this case here, if we had the 3 letters AGT for adenine, guanine, and thymine at a particular position on a DNA strand, it will result in the placement of the amino acid called serine at the corresponding position of the polypeptide, which will be produced later. So every 3-letter combo is going to produce a particular amino acid. So how do we decipher the code? Of the 64 triplets, 61 code for amino acids, but 3 of them are triplets that serve as stop signals to end translation. There's also a start signal as well. The codon AUG codes for the amino acid methionine, and that will start a sequence of a gene. So as we look at the process of translation later, you're going to notice that all of the polypeptides seem to start with this amino acid methionine, and the reason is because it is the start codon. The genetic code is what we call degenerate or redundant. And what that means is that you can have more than one codon coding for the same amino acid. Remember, we have 61 actual triplets that actually code for amino acids, and we have 20 amino acids. So what that means is we're going to have some of the codings, there will be different codings, but they'll code for the same one, and that will make up for all the extra combinations that we have. What we could also say on the flip side of that, though, is that the genetic code is also unambiguous. And what that means is that every 3-letter code is going to code for exactly one thing, and it's usually an amino acid. So no one 3-letter combo can code for 2 different amino acids. That can't happen. So why is it that we have more than one 3-letter combination, triplet, being able to code for the same amino acid? Well, one of the things you can see over here on the right is that it protects against the harmful effects of mutations. One of the things that can happen is occasionally one of the letters will be switched inside of a triplet. And wouldn't it be beneficial to an organism if it didn't completely shut the organism down, but instead there was a little room for error in something as simple as one base, so the organism could continue to live its healthful life, and the mutation isn't so bad as to cause something extreme in the outcome. So we'll learn a word a little bit later that goes with this that allows for a little bit of sway in the genetic code, but we can still nail down the proper amino acid. This is an mRNA chart, and what we can see, first let me explain how it works. We have the first mRNA base, so the first letter is going to be along this left-hand side right here. And you can see our 4 letters for RNA. We have G, we have A, we have C, and we have U. Okay, because an mRNA transcript is always going to start with one of the 4. And we said that the genetic code was written in triplets or codons. And those codons have 3 letters. So the second letter, whatever it may be, is found here on the top. U, C, A, and G. And the third letter that we have in our codon is found over here on the left. And what we're going to do is whenever we get a codon, we can then decipher what amino acid it would code for by looking at this chart, finding the first letter, crossing it with the second letter, and crossing it with a third letter. Okay, so a couple of things that I point out on this mRNA chart is the degeneracy or redundancy idea we were talking about a minute ago. And we said it can protect against mutations because check out this these three different codons here, which can all code for the amino acid isoleucine, AUU, AUC, or AUA. All of them code for isoleucine. So what that means is if the last letter, this one right here, gets changed to something else, we still have the right amino acid placed in our polypeptide chain that's growing. Okay, so let's go ahead and check out some letter combos and see what they may code for. Let's look at AUG first. So we have the three-letter code AUG. What we do is, since the first letter's over here, we go to A. Here's U, so it's going to meet in this box with those others we were just talking about. And then G is down here, and we cross this over to here, and what do you know? We land on our start codon, which codes for met or methionine. Let's say, though, that your next amino acid was GGG. So again, we scan down our first mRNA code. Here's letter, here's G. So we're going to be in this row here. Here is our second letter, G again, and that means that we're going to be in this box right here when we cross the 2. And the third letter is G as well, which gives us this code. And you can see that all 4 of these codons code for the amino acid glycine. So GGG would represent glycine in a growing polypeptide chain. We're now going to talk about the actual process of transcription. What goes down to allow that mRNA molecule to be built? So during transcription, one of the 2 DNA strands, and we're going to call that the template strand, will provide the sequence of nucleotides that will be complemented in an RNA transcript. RNA synthesis is catalyzed by RNA polymerase, which pries the DNA strands apart and hooks together the RNA nucleotides. Now, when we learned about DNA replication, we learned there was a whole cast of players that allowed that to happen. We had the enzyme helicase. We had 2 different DNA polymerase we talked about. We talked about DNA ligase, single-strand binding proteins. Not as much goes on here as we copy segments of DNA, but RNA polymerase is our major player that we're going to focus on as this process happens. So it pries the DNA strands apart, hooks the new RNA nucleotides on, and the way it does so is by simple complementary base pairing, except now we're using the RNA rules versus the DNA rules. So A will code for uracil. If G pops up, we know it codes for cytosine. Thymine will code for adenine, and cytosine will code for guanine. So there's our 4 connections. This big picture here shows the process occurring. Now, what is happening here? What is all this stuff? Let's go through these things one at a time. First, the strand that's being copied in this one here is our template strand, and you can see here it's this darker blue colored, and you can hopefully see that the dark blue was connected to the light blue a minute ago before it was pried apart, and the one that's being copied is this one right here, template strand of DNA. Now, that means that we have a non-template strand, which we have up here at the top, and in the lighter blue color. Now, the thing that's prying this DNA apart is this big white thing that we're looking at right in the middle, RNA polymerase. So this big protein right here is prying it apart, and it's allowing for what you can see here as new nucleotides entering into the area and being connected on to the template strand. Direction of transcription, as we can see here, is going to the right. So what we would have, if this was our start of our transcript, eventually we're going to get to the end of it once that particular transcript is done copying. So we have the copying going on in a 5' to a 3' direction. Okay. Now, this 5' to 3' direction, the code here is going to be identical to the non-template strand up here, except, of course, we have U in it. So essentially, our template strand, the one on the bottom here, is being copied from a 3' to a 5' direction, but our mRNA, which is this red thing, is being copied in a 5' to 3' direction in itself. That can get a little bit confusing when you're trying to sort through, okay, it's anti-parallel, 5' to 3', 3' to 5'. That can be a little bit confusing, so take your time with it. Sort out in your mind what direction things are happening in. The fact that your mRNA completely matches your non-template strand except has uracil in it. Next, let's take a big step back and let's take a look at what's going on here in terms of a longer piece of DNA. What is all happening? So here you can see that this DNA segment is made of 3 different genes. Which one we're going to focus on right now, of course, is gene 1 that you can see here. This little box is opening up to here. And here is the sequence that essentially is going to need to be copied. So this is going to be referred to as our DNA template. And up here, this is our DNA template strand. So it's going to be the top. It will separate due to RNA polymerase. It's going to go in there and wedge these two apart. We'll then see transcription occur, where we are now forming a copy of essentially this particular stretch of DNA from here to here. So it gets pried apart, and when it's pried apart, we have these new nitrogenous bases of mRNA being added in, and you can see that these two strands here are identical except for the fact, again, that RNA has U's instead of T's. After that's built, that's your mRNA, you're then going to eventually get to the translation process, which is going to translate each of these 3-letter segments here into actual amino acids in a growing polypeptide. Taking another step back, looking at the transcription unit, which is the entire stretch of DNA that's going to be copied, we can look at it in terms of a few different steps. We could call it initiation, would be the starting of the process. And you can see that mRNA polymerase is up here, and it's responding to something, a stretch called a promoter, which is going to kind of turn on the copying process. And you can see that the DNA is currently being spread apart here, and you now have an RNA transcript that's being built. So this RNA transcript is going to get longer. And that's going to happen during elongation. So as this moves forward, this RNA polymerase, it will continue to wedge out the DNA, allowing it to open up a little bit, and it will allow that copy to get longer and longer. And we said the copying goes on from the 3' to the 5' end on the template strand, but the copy itself of mRNA is actually built in a 5' to 3' direction, an anti-parallel direction to the strand it's copying. And eventually it's going to get to the end of the stretch that it needs to copy, where then the DNA goes back to the way it was before, and you now have a completed mRNA transcript here, and that would happen during termination of that transcription process. Up here I just put a couple of interesting facts that how amazing the machine is. Transcription progresses at a rate of 40 nucleotides per second in humans. That's extremely fast. So 40 of these little these nitrogenous bases here, the nucleotides that are adding, are going on per second all the time in every cell of your body. Another thing that we need to point out is that genes can be transcribed simultaneously by several RNA polymerases. So here we just have one working on the DNA. But what if there was another RNA polymerase right behind it and it started copying behind it? That's what we're talking about here. So you can have multiple copies of the same message being written onto mRNA and then translated into a protein elsewhere in the cell. So we also mentioned that in a bacterial cell, from the mRNA transcript being built, it's then going to go find a ribosome where translation will occur. But in a eukaryotic cell, we got to have a little bit of modification go down on a transcript before that can happen. So what happens is on each end of those mRNAs, and we'll call it a pre-mRNA because it's not out of the nucleus yet, it's going to be modified a little bit. The 5' end of it is going to receive a modified nucleotide cap. And that's what you see down here with these phosphates that seem to be added in this yellow portion of your mRNA. The other end, we get what's called a poly-A tail, which is just a sequence of a whole bunch of adenine over and over and over. And what that does is it's going to provide some protection to this message that will be decoded in a little bit in the cytoplasm. What purpose do they serve? You can see down at the bottom, they seem to facilitate the export of mRNA, gets it out of the nucleus and on its way to translation, protects mRNA from hydrolytic enzymes. So if anything is eyeballing this mRNA to possibly break it down and use it for spare parts, it's not going to happen because it now has a little bit of protection on it if it gets nipped. They help ribosomes attach to the 5' end. So when we talk about the translation process, this is going to allow it to adhere to the translation complex a little bit easier than if it wasn't there. Another thing that needs to happen before this particular message is ready for translation is something called RNA splicing. So the reason we have to splice it is because there's long non-coding stretches of nucleotides that lie between the coding regions. So the non-coding regions are called intervening sequences, or much simpler, introns. Introns need to be cut out because they're not actually coding for anything. And what we want translated eventually is a meaningful message without any extras. So the extras, the sequences that need to be copied and not the extras are actually the introns. What gets expressed by way of a protein is actually called an exon. So I like to think of exons being expressed. They both start with the EX letters, and you can think of that in your mind when you're trying to remember which one actually gets turned into the working polypeptide and which one gets cut out. So eventually exons will get translated where introns don't. And the whole process is called RNA splicing, as you can see in the heading of the slide. It removes introns and joins those exons, creating an mRNA molecule with a continuous coding sequence. As I make these screencasts, sometimes words get jumbled in my mouth, so much to the point where, I don't want to leave that on the video. So when I get to the editing process, it allows me to look and replay what I said, and I can cut out chunks of it and splice together what's left over. Or if I pause for 10 seconds and I don't want, the viewer watching 10 seconds of silence, I can cut that, pull it out, and then stick the rest together. So just like when you're editing film or audio tape, it's the same thing going on here with RNA. We can splice out what we don't need. So in eukaryotes, RNA splicing is carried out by something called a spliceosome. And right here in this copy, this pre-mRNA, you see that the 5' cap and the poly-A tail have been added already. But what we don't need going to the ribosome are these sections here, this intron and that intron, because they don't code for anything. What we do want is all this stuff, and this stuff. We want them because they're going to be expressed. So what happens is that these introns are going to be cut out and the exons will be spliced together by a spliceosome, which you'll see in a minute. Now, if I explain what a spliceosome is, it's kind of like head-blowing, mind-blowing things going on here. But I'll get through it. It says, spliceosomes consist of a variety of proteins and several small nuclear ribonucleic proteins, better known lovingly as SNRPs. They recognize splice sites. Also utilized in the cutting out of introns are going to be ribosomes, which are RNA molecules that have catalytic activity. So let's check out and see what a spliceosome actually looks like. So here we have an mRNA transcript. Intron is this rather large portion in the center. And here we have a protein that's bonded to a small nuclear RNA, or together those things are better known as a SNRP. And it's going to come together with other proteins and it's going to form what we call a spliceosome, which you see here. You can also see that we now have bent the mRNA molecule into this U shape, and when we have two cut sites, one's going to be here and one's going to be here. The spliceosome is going to take care of that. You can see that over here we kind of have this cutout intron portion, the light pink section. It's kind of in this lariat-like structure. And what's left over is the two dark red portions of exon 1 and exon 2 that we wanted the whole time. So we got rid of the extra, we put it back together, and now it's ready to move on and be translated into a working polypeptide. Our process number 2 is called translation. A cell will translate an mRNA message into protein with the help of transfer RNA, the second class of RNA, which we simply called a tRNA. Molecules of tRNA, however, are not identical. Each one has what's called an anticodon on one side. And the anticodon is going to base pair with a complementary codon on the mRNA. So eventually there will be a meeting between the two, the mRNA that we've been talking about and a tRNA molecule. And there's going to be a binding moment where complementary base pairs are going to find each other from the codon of RNA, mRNA, to the anticodon of tRNA. Each tRNA will also carry on its other end an amino acid. So hopefully you can start to see how this is coming together. That it's going to bring an amino acid in, and by way of the codon and the specific matching, we're then going to start producing a working polypeptide chain. tRNAs consist of a single RNA strand that's about 80 nucleotides long. If you flatten it into one plane, it's going to have a clover leaf-like shape. You'll often see this kind of shape when you're looking in textbooks. But we know that in the real world, proteins are not flat. Instead, they're three-dimensional. And what's going to happen is it's going to fold itself into almost like an L-like structure, an upside down and backwards L, which you can see down here. And you can see on one end, you have an amino acid attachment site, and on the other end, way down here, you have the anticodon, which we said a minute ago was the three-letter combo that matches up a complementary base pairs to the mRNA's codon. So the amino acid attachment site's on one end, the anticodon is on the other end. For translation to be accurate, it's going to require two things to go down. Number one, a correct match between tRNA and an amino acid, which is going to be done by the enzyme aminoacyl tRNA synthetase. Wow, that's a mouthful, okay? But it is what it is, so we'll just acknowledge that, okay? So what we see happening here is the amino acid, and you're going to see that it has a specific shape, and it's going to fit into the enzyme. Remember, lock and key of enzymes. And you also see ATP, so it's going to take some power in order for this to happen. Now, once the amino acid nestles into this active site here, and ATP is then going to be spent to get it in there, to provide its conformational change, we then see an adenosine monophosphate left over that's still attached to your amino acid, which is still nestled into your enzyme. Then we have a tRNA float in, and what it's going to do then is it's going to then bind with the particular amino acid that we see here, and it seems to kind of chemically rearrange to get rid of the adenosine monophosphate, and now what's left over is the actual part that we need, which is a charged tRNA. A proper amino acid on the top, along with the proper anticodon on the bottom, and this thing is ready to go, ready to add this amino acid to a growing chain. The second thing required is going to be a correct match between the tRNA anticodon and the mRNA codon. So where that occurs, we can see right here, there's a lot of action going on here, but the mRNA's UUU codon is being adhered to by the AAA anticodon of this tRNA. So this thing here again is a tRNA, and on one end is the anticodon, which we have right here, and on the other end is the amino acid, which we can see right here is phenylalanine. There we go. And it is currently part of a growing chain. Remember we said that there is some flexible pairing at the third base of a codon. That's also called wobble. It allows some tRNAs to bind to more than one codon. Remember, some of the amino acids have 4 different letter combinations, and they all code for the same amino acid. So it's a little bit more efficient to use that system for tRNA. Ribosomes are ultimately the location of translation. So what they're going to do is they're going to facilitate coupling of tRNA anticodons to mRNA codons during protein synthesis. The ribosomes are made up of 2 units. There's going to be a large unit and a small unit, and they're made of proteins and ribosomal RNA or rRNA that are produced not in the nucleus but the nucleolus. Remember, that dark spot in the nucleus actually has a function, and that's it. It's ribosome production. Now, they're going to move out of the nuclear pores on their way out of the nucleus. They're either going to go to the free area, which is going to be the cytoplasm, or they're going to be bound. They're bound ribosomes. Of course, we saw those back when we were studying the cell, and we mentioned endoplasmic reticulum, and particularly rough ER. That's where we found those. So those are the place of protein synthesis. So they're either going to happen attached to a membrane, or they're going to happen freely floating in the cytoplasm. If we check out the anatomy of a ribosome, we're going to notice it has 3 binding sites for tRNA. The first site we'll mention is called the P site. The P site holds the tRNA that carries the growing polypeptide chain. And you can see directly in the center here, we have the P site right there, and if we look at the computer 3-dimensional model, it's also in the middle, slightly off to the side. That's unimportant for now. We're learning the mechanisms of how this thing works. So the next site we'll mention is the A site, and the A site is right here. It's the first site for much of this process, and it holds the tRNA that will carry the next amino acid to be added to the chain. So really the P starts it off, and then A will continue to fill up with all the new amino acids that need to be added to the growing chain. And the last site is called the E site. I like to think of it as the exit site. And this is where discharged tRNAs will leave the ribosome. So once they've lost their amino acid, they're then going to be discarded to be used again, to be charged up with that fancy enzyme name. So all is not over for these tRNAs. They can be reused. So let's talk about how this occurs. And if you're trying to remember the sequence of them, if you go from right to left, it spells APE. If you go from left to right, it's EPA. And we all know Environmental Protection Agency. So either way you want to remember it, hopefully you can remember the order of them by either one of those acronyms. So how do we build a polypeptide? There's 3 key stages of translation. First, initiation. Something needs to get this process going. We'll take a look at that very quickly. Second, elongation. Once it gets going, how do we build every single amino acid down the way? How does it work? We'll look at that as well. And lastly, termination. What determines when this thing stops? And that will be our last stop as we discuss this process. During initiation, what we'll see is an mRNA, a tRNA, and the 2 ribosomal subunits get together. Let's kind of break that down for a second. So number 1, it says a small ribosomal subunit binds with mRNA and a special initiator tRNA. The small subunit locates the starting codon AUG. So we mentioned that there was a start codon that coded for methionine, which you can see here. And the process really gets going when the tRNA's anticodon meets up with the codon for that. Remember, before your AUG, you might have a 5' cap on your mRNA, and that's fine. But these will find each other, as well as the bottom piece, the small ribosomal subunit of the ribosome, and it will all connect together. And it will look like this at the connection point. A little bit of energy is spent. As we know, GTP is a lot like ATP. And what's going to happen is we are then going to allow the large ribosomal subunit to connect on to the whole thing. So now we have almost what's called a translation complex, which you can see down here. Translation, initiation, complex. The large subunit attaches to the small subunit with the mRNA, the red strip here, sandwiched in between. The tRNA moves into the P site. A site open. All right, so now we've kicked it off. We are all ready to go. So the next thing that needs to happen is elongation. We need to go from that first amino acid methionine into additional amino acids to build a polypeptide. So they're going to be added one by one. And if we broke this part down, it probably has 3 sub-steps that are worth mentioning: codon recognition, peptide bond formation, and translocation. If we look at codon recognition, we're going to notice some things happening here. We're going to notice that currently we have our tRNA present in the P site and the A site is open. We have an anticodon here. We have a codon here. They are going to find one another chemically, and this tRNA is bringing the proper amino acid into the mix. And that would be codon recognition, the complementary base pairing of mRNA to tRNA. Now, we notice that the tRNA has nestled into the A site, and the growing chain that's currently sitting on the tRNA and the P site, you can see that there is a shift over. If you remember what links amino acids together, it was peptide bonds. So what we're going to have happen is we're going to have this growing chain right here shift over to the A site by way of a peptide bond formation, which is happening right here. And now we're going to see a shift in both of these. It's going to be called a translocation. So P is going to go to E and A is going to go to P, which you see right here. Now, because the tRNA from the P site is now empty, it's already added its amino acid, it can be released. So it's going to be released from the E site, and now the chain with one more amino acid has translocated and moved to the P site, and again, A is open. So now the next codon that's exposed here is going to bind with the next anticodon from a tRNA, and it's going to happen very rapidly. If you get a chance, pause the video, write down this website right here, and go check it out. It's a very nice animation showing this process in action. Amino acids will be continually added in that way until you reach a particular sequence of letters, codons that code for stop signals. So those 3-letter combinations are UAG, UAA, or UGA. If those 3 letters show up in a codon, what's next is the termination of the translation. What's going to happen is we're going to see that the A site will accept a protein, not an amino acid, but a release factor will come into play. So our release factor now, instead of a tRNA, is what's present in that location on the A site. When the release factor binds, instead of adding an amino acid, there's going to be a water molecule added. And that water molecule is then going to allow this to happen. So everything will come apart. Translation has come to an end by way of adding a molecule. Both subunits of the ribosome are released. The mRNA is released. The release factor is released. And we're ready for a new message somewhere else within the cell. We mentioned earlier that sometimes a number of ribosomes can translate a single mRNA simultaneously. So what if we needed this particular protein and we need a lot of it? If that was the case, we may see a number of ribosomes. In this case here, this would be number 1, 2, 3, 4. And the reason I called that number 1 is because it's already completed building it. Number 2 is still working on it. Number 3 just got started. Number 4 is really just got started. So they kind of look like beads on a string, and that's how they're referred to. And we call that a polyribosome. So if you need a lot of this particular polypeptide right here, you might see a lot of translation going on a particular strand, and you can see that right here in that electron micrograph. So many copies of a polypeptide can be produced quickly that way. We mentioned, looks like beads on a string. Now, the last thing that needs to happen in many cases is you've built a polypeptide and it's kind of floating around in the cytoplasm, and it might not be quite what it needs to be yet. So there's helpers around within the cellular environment, and we call those chaperonins. So occasionally, if the polypeptide has not coiled and formed the proper structure, what we see is a chaperonin, which you can see down here, will come in and help out. And what that's going to do is allow it to go into a bigger protein structure, be folded up properly, and then released to go do whatever it needs to do. So if we take a look at what a chaperonin actually looks like, it would be this big, garbage can looking thing here. Here is our polypeptide that hasn't been folded quite right yet. And what it's going to do, it's going to enter the chaperonin. There's a little cap that winds up going on the top here. And then upon folding properly within this chaperonin protein, it will be released. And here you can see it's a lot more condensed. The folding's a little bit different than it was before. The cap releases off. The protein now goes and does whatever it needs to do. And the chaperonin can be used again for proper folding if necessary. So these are the two big processes that aid the central dogma of biology. We go from DNA to RNA to protein. From DNA to RNA, we use transcription, and from RNA to protein, we use translation. Okay? These topics take a lot of time, a lot of energy, and focus. So make sure you read your book. Make sure you ask your teacher questions if you have them, and I'll talk to you next time. Good luck.